Hello everyone, I'm Dr. Fuller and welcome to your first video lecture of BIOL 251. Today we'll take a couple of minutes and talk about the cell cycle. Now this may be a review for some of you or it may be an introduction for you, but either way it's very important that we cover it early in the semester because it really does kind of lay the foundation for everything that we'll talk about going forward. So it's good to have the knowledge of the cell cycle kind of running in the background of your brain, so that as we go through, when we start to talk about DNA replication and gene expression, what we're really talking about is what's happening in the cell leading up to cellular replication. Okay, so just to orient ourselves around the schematic that we see here, so you see this circular illustration, and this represents the cell cycle. You also notice these directional arrows embedded in the illustration. So what these are showing us is that the cell cycle here is envisioned as this kind of clockwise process that keeps repeating itself over and over again. Okay, now that we've gotten that out of the way, let's get to it. So the cell cycle refers to a series of events that describe the metabolic processes of growth and replication in the cells. Most of the cell cycle is spent in the living phase, known as interphase. Interphase itself is broken down into three distinct phases. First we have G1, then we have synthesis or S phase, and then there's G2. And after those three phases, the cell moves into mitosis, which is the focus of Module 1.3. So let's go back to G1. G1 is the phase of growth when the cell is gathering resources to live and grow. So sort of a resource gathering phase. After the cell reaches a certain size and it has gathered enough raw materials, it moves through to this checkpoint. So you see I have a little stop sign here. Checkpoint is a stopping point. At the checkpoint, the cell uses biochemical markers to decide if G1 was successful and the cell has all the materials it needs to move into the next phase. If so, the cell cycle will progress into synthesis phase. If not, the cell exits into this G0, which is kind of like a waiting room. So that may be waiting for more resources or decided to start the process all over again. But let's assume that G1 is successful, then the cell moves into synthesis phase or S phase. During S phase, the amount of DNA in the nucleus is doubled and copied exactly in preparation for cell division. Now, when DNA synthesis is complete, the cell continues on to the second growth phase called G2 or GAP2. This is another opportunity for the cell that now has double the DNA than when it started to take a little bit more of a moment and grow to accommodate the newly acquired, newly synthesized DNA. At the end of G2, we see another checkpoint here. So there's another stopping point. At this checkpoint, the cellular machinery is checking for damage. So it's making sure that all the DNA is synthesized in synthesis phase, and during the second growth phase, there was no damage to the DNA, and the DNA is, through a quality control step, ready to go for cell division. Okay, so if all the conditions are favorable when the cell reaches the G2M checkpoint, then the cell continues on to mitosis. And so we'll spend module 1.3 talking about mitosis, but for now, that's the cell cycle. So what are the takeaways? The cell cycle is, like it says, it's a circular process that keeps repeating over and over so that the cell can reproduce. It has really two major phases, interphase and mitosis. And within interphase, there are three subphases, G1, S, and G2 phases. And we'll remember these checkpoints, which are areas, times that the cell can stop and make sure that the cell cycle has been successful up to that point. Okay, so now what you will do is move on to the next activity in module 1.2 in preparation for module 1.3, which is mitosis. Okay, catch you later.